The following content is provided under a Creative Commons license. Your support will help MIT OpenCourseware continue to offer high-quality educational resources for free. To make a donation or view additional materials from hundreds of MIT courses, visit MIT OpenCourseware at ocw.mit.edu.
What we're going to do today is just discuss a few aspects of cross-linking. So we decided it was important to introduce this within recitations this year because cross-linking comes up time and time again. And there's different ways to do this and different strengths and limitations to different approaches. So I guess in just thinking about this, what is cross-linking? So if you say, oh, I'm going to use a cross-linker for my experiment, what does that mean?
Forming a covalent linkage between two molecules of study.
Yeah, so there's going to be formation of some sort of covalent linkage between two, or maybe more, right? Because some cross-linkers can have more than two reactive groups, okay, of study, right? So we're chemically joining two or more molecules. So why might we want to do this? What are possible applications?
Study protein-protein interactions.
Yeah, so that's one. So protein-protein interactions, right? And that could be identifying unknown protein-protein interactions, or maybe you know two proteins interact, but you don't know how, right? And you decide to use cross-linking as a way to probe that. So how might cross-linking help with studying a known protein-protein interaction?
Figure out or start to get an idea of where the proteins are actually interacting, or which residues might be important.
Yeah.
And then it could allow you to isolate them, right? Cross-linking.
So if you don't know of it's an unknown protein-protein interaction,
Right, so maybe there's an unknown one,
Right.
and you fish that out because a cross-linker was used, right? And that you know what one of them are. Or maybe, say, we know, right, that these two proteins interact somehow, but we don't know how, right? So, is it at an interface on this side, right, versus maybe the other side, right, versus maybe behind the board, et cetera, right? And so there's many ways to study protein-protein interaction, and really how I'll present cross-linking today is in the context of this particular application, but there are many others, right? But if we just think, we've seen a lot of protein-protein interactions in this course, right? So just even today, ClipXP is an example, right? We saw protein nucleotide interaction with the ribosome. GrowEL, GrowES is an example of protein-protein interactions, right? And they've been studied by many other methods, like crystallography, for instance. But sometimes maybe it's not possible to get a structure, right? And you want to define an interaction surface or know exactly what residues are important, right? So here, say it's protein-protein. But that could be generalized to any other type of biomolecule like RNA, DNA, right? What about a single protein, right? So can you use cross-linking to learn more about tertiary structure or quaternary structure, right? So imagine, for instance, rather than two separate proteins, right, we have one protein where there's some flexible linker, and we have reasons to believe these different domains interact, right? But how do they interact? Again, is it something like, this undergoes some conformational change and they're like this, versus other possibilities here. So what about just other applications of cross-linking chemistry before we look at some examples of molecules? So we can, capture and identify binding partners, as Lindsay indicated. We can study known interactions. Where else could this come up? Right, and although it wasn't defined in this way, we've seen certain technology that takes advantage of cross-linking chemistry often.
Within the realm of biological things, it's used for, if you want to attend a functional groups, so like biotinylation or general bioconjugate chemistry for like drug delivery.
Yeah. Right, so general, exactly, general bioconjugate or conjugation chemistry, right? So maybe you want to attach a tag to a purified protein, maybe you want to modify an antibody. Similar chemistry can be employed. And likewise, even like from application standpoint, immobilization. So say you need to make your own resin to do some sort of affinity chromatography and you want to attach a protein or an antibody to that. You can use the types of chemistry shown here. So we're going to talk about a few different types of crosslinker and the chemistry and pros and cons. And just as a general overview, how I'll describe types. Okay, so we just heard the word homo-bifunctional. So homo-bifunctional versus hetero-bifunctional. Okay, and this refers to the reactive groups. So we need to talk about what types of chemistry is going to be used to do cross-linking. So this refers to reactive groups. And then another classification will be nonspecific versus specific. And so this doesn't refer to, say, the chemical reaction between the crosslinker and whatever it's hitting, but rather whether or not the cross-linking reagent is site-specifically attached to a protein or biomolecule of interest or not. Okay? So this, okay. So if we just think about this nonspecific versus specific, if we want to attach a crosslinker at some specific site in a protein, how can we do that? So think back to the ribosome discussion, right, where unnatural amino acid incorporation was attached. Was not attached, but was introduced, right? So that's one possibility. If you have an aminoacyl tRNA synthetase and a tRNA that can allow some sort of crosslinker to be introduced site-specifically, and it works for your experimental situation, you can do that, right? So we saw benzophenone, which is a crosslinker in the evolution of that orthogonal ribosome riboX. But let's say you can't do that, right? So for instance, as far as I know, there's no tRNA AARS pair for benzophenone in a eukaryotic cell, right? Or maybe in some circumstance. What is something just using standard biochemistry you could do? So what type of residues can be modified in a protein? Yeah, so cysteine, lysine, these are common side chains that are modified. And what would you say is more typically employed if you want to introduce a site-specific modification using chemistry? Cysteine, right? So if you have an individual cysteine that's in the protein, or maybe you use site-directed mutagenesis, right? You know where that cysteine is, and then you can modify it with some reagent there. We'll come back to that in a minute. So in terms of reactive groups then on the protein, we can think about lysines, right? We have the epsilon amino group, cysteines, we have the thiol. What do we need to think about for our chemistry when thinking about these types of side chains and wanting to do a reaction? So under what conditions do we have a good nucleophile? Pardon? Yeah, so we need to think about the basicity, right, the pKa of these groups, right? That's very key here for that. What else do we need to think about? You know, what other factors might govern reactivity? Just thinking broadly. So pKa, right? For your amine, it will be type of amine, right? For a cysteine, redox will play a role, right? You can't have your cysteine in a disulfide. It needs to be the free thiol form, right? So these are all things to keep in mind here. So Alex has used a homo-bifunctional crosslinker. Why did you use a homo-bifunctional crosslinker?
It was to like stabilize a nanoparticle.
To stabilize a nanoparticle. Okay, so very different type of application here. So, okay, that's fine. So, yeah, we're not doing much with nanoparticles here. But let's say we want to use, you know, a nonspecific homo-bifunctional, so this was nonspecific crosslinker to look at some protein-protein interaction, right? So if we just suppose, for instance, we have some protein A, and we think it interacts somehow with protein B, how can we use crosslinkers to study this? So let's take a look at an example of a homo-bifunctional crosslinker in terms of design. Here. So this one will be amine reactive. And its name is DSS here. Okay. So effectively, if we want to dissect this structure into different components, what do we have? Yeah, so we have two reactive groups, or leaving group, separated by a linker. And in this case, we have two NHS, or 6-cinnamyl esters, right, that are amine reactive. So what's the product of reacting an alpha amino group or a lysine epsilon amino group with an NHS ester? What do we get? An amide, right? We get an amide bond. And then we have this linker or spacer region, okay, here. So two amine reactive groups and a linker or spacer. And in this particular case, this linker or spacer is about 11 angstroms. And it's flexible here. And it's stable, right, and cannot be cleaved here. So in the case of Alex's project, this was used to stabilize a nanoparticle. And the nanoparticle, did you have a pure nanoparticle? Or was this in a very complicated mixture?
It's like very modest.
Okay. So what's going to happen if this reagent, say, is added to cell lysate? What are you going to get?
Random cross-linking with a bunch of different lysate proteins or cytosolic proteins.
Yeah, so there's a high likelihood, right, of a lot of different cross-links, right? So potentially a big mess, right? High likelihood, right? Because you have no control over where these reactive groups are going to hit, right? And do most proteins have lysine residues? Yeah, right. Do all proteins have an alpha amino group? Yeah, right. So, well, some might be, modified. But anyhow, you have very little control with this type of reagent. So then the question is, if you use it, how are you going to fish out your desired protein-protein interaction, right? Or even if you're working with two purified proteins and they have multiple lysines, you can end up getting multiple crosslinks, right? So maybe that's helpful for initially identifying that an interaction exists, but in terms of getting more detailed information, in terms of how do these actually interact, that may be tough here. Okay, so easy to come by, but potential complications. Just in terms of thinking about this in the linker, why is it important to think about the linker in your choice of some reagent here? So what properties does the linker give?
There's length of the linker, and I guess its flexibility would determine what the crosslinks, like how close the two proteins have to be in space for this to be able to occur.
Mm-hmm. Yeah, so there's some constraints imposed by the linker in terms of, how close together or far away are groups that react. What else comes with the linker? How does it affect the properties of the molecule? Alex?
I was going to say it can dictate how likely you get cross-linking on the same molecule between like two amines.
Mm-hmm.
Like if you make it short enough so that it can't reach the next lysine or something,
Mm-hmm.
then you can prevent intramolecular cross-linking.
Yeah, may be able to. So what's an inherent property of a molecule? Yeah.
It might affect solubility.
Yeah, right. It may affect solubility, right? So linkers can be, this is, a bunch of CH2 groups, relatively hydrophobic, right? There can be more hydrophilic linkers or other strategies. And then the question is, does that matter, right? Does the solubility properties work with your experiment or not, right? But imagine if you want to do cross-linking in a live cell, you need that cross-linker to get into the cell, right? So you need to think about membrane, permeability, and what happens after that. Here, so the linker is another critical aspect, and so if you're ever working with a cross-linker, that's something you want to think about in addition to what types of side chains or what types of biomolecules you want to modify. So let's look at an example of a hetero bifunctional linker. And this one, oh, it's not linker. Well, it is, cross-linker. Okay, here for this. So this one will have a different type of spacer group. So it'll be with a cyclohexyl. So what do we have in this case? Steve? Yeah.
So you have an NHS ester and also a malamide.
Yeah.
And then the sulfonate group probably helps with solubility.
Yeah, right. So there's a bunch of interesting aspects to this molecule. So we have the NHS ester to react with an amine. Right here we have a malamide, which will react with thiols. So hetero-bifunctional because there's two different reactive groups for different types of side chains. And then, as Steve mentioned, we have this group here, right? And so this is to improve water solubility. Okay, and then what do we have in this linker region? Oh. Yeah, and what does that give? Yeah, exactly, like cyclohexyl, right? Think about chair conformation rather than what I have drawn here, but it will give a more rigid linker and also shorter than what we see up here. So this is on the order of 8 angstroms. Okay. So how might this molecule be used? What could you do with it that you can't do with this one?
Cross-link cysteine and lysine.
Yeah, well, that's the first point, right? You can have 2 different groups, right? One end will react with a cysteine, one with some lysine, right? So is this specific or non-specific or both?
Probably depends on the context.
Yeah, right, could depend on the context, right? And then from the standpoint of specific cross-linking, which I would argue is a better use of this compound, what can you do, right? You can have, just imagine you have some protein of interest, and maybe you want to label it here, right? And you have some side chain, so site-directed mutagenesis, to put in a cysteine, right? And then you can modify that there, right, such that you have cross-linking reagent, right? Here. Right, and then you can imagine, you know, whatever your experiment is here. So again, thinking about using this compound in, say, a complicated mixture like a cell lysate, right? You want to see if there's any binding partners or whatever. What's the limitation in terms of reactivity of this amine group that you'd use in that second step? Where do you lack control?
You still can't control for the alpha, for the N-terminal reaction, right?
What do you mean by that?
Well,
So
if the N-terminus is free, then would you have comparable reactivity between the N-terminus and, for example, your desired lysine if you were trying to do?
Okay, so that could be an issue. Right, so do lysines and N-terminal alpha amino groups have different reactivity? Do they have different pKa's, and is that something you could control? Maybe, maybe not, but more broadly than that, so you have an issue that it will react, let's say, with any amine, right? Can you control when it reacts?
To some extent, probably with pH.
Mm-hmm. So what are you thinking?
Well, like if you,
Right, so if you think about just experimental design, right, and say you were to try to use pH to control reactivity, and I'm defining this broadly, reacting with any, you know, amino group, so we're not going to try to do something to selectively label one, right? This is reactive. It will react, right? So would you pH change your whole buffer or pH change the cell lysate and then switch to turn on reactivity? Probably not. Probably not, right? That I'd say is not very likely, right? So the issue I'm getting at here is that you have little temporal control or spatial control of an NHS ester, right? It will react with an amine provided, you know, your conditions are appropriate. So just getting back to this pH issue and a little digression, if you want to use something like an NHS ester, you know, in a test tube experiment, what you need to think about beyond pH, right? Yeah, so what you need to think about with the buffer.
You don't have tris, well, tris buffer can cross-react with these, so you might want to use like a phosphate buffer, something that doesn't.
So this is a key point. You need to think about cross-reactivity with the buffer. So if you have tris buffer, you have amine. If you have a buffer that's like glycine, there's amine, right? And your buffer concentration is, in most instances, much higher than whatever the concentration is of the molecule you want to actually modify, right? If you think about 10 millimolar tris or 75 millimolar tris, right, compared to micromolar or nanomolar of some protein. So you need to have a buffer that's not reactive, right? You need to have an appropriate pKa. Those are important considerations. You need to know that your reagent is good, appropriate pH. What about the thiol here, what you need to think about if you're doing a test tube experiment and want to modify a thiol with a malayamide or something else like iodoacetamide that we saw last time.
Buffers we need to avoid like DTT or.
Yeah, so what's DTT? Right, yeah, or BME beta mercaptoethanol, right? Even before that step, what do you need to make sure? So what if there's, multiple cysteines?
That they're not disulfide.
Yeah, right, so either inter or intramolecular, right? So if a reducing agent's added and the reducing agent is thiol-based, again, you're going to have much more reducing agent than your protein of interest, right? So you don't want your thiol reactive probe to react with the reducing agent in the buffer here, okay? And so that needs to be removed. And then if you remove it, you need to ask, does the thiol stay reduced, right? Or is it susceptible to air oxidation, right? So these are just all practical considerations to keep in mind. Like if a reaction doesn't work, why doesn't it work? And was it something that wasn't right with the buffers there? Okay, so back to this issue of, not having much control about, timing control for reactivity of these types of groups. What could be done to overcome that? So what other types of crosslinkers are out there? Yeah, Photo-Reactive crosslinkers. So what's the idea here?
Control formation of the activated state using the appropriate wavelength of light.
Yeah, so what do we have and what can we do? So just the first point to make, right, is that we want to think about specific labeling here. Okay, so we can attach site specifically to a protein or some other biomolecule, right? Maybe it's by cysteine modification with something like a malayamide. Maybe it's unnatural amino acid incorporation, okay? And it's chemically inert. Okay, until irradiated. Okay, and so basically irradiating this photoreactive crosslinker will activate the photoreactive group, and then you get crosslinking. Okay, so this type of approach is often used to capture binding partners. It can be used in the test tube or in cells. What are the types of photoreactive crosslinkers? Here. Yep.
Aryl azides?
Yeah, so aryl azides are one type. Okay, what's one we saw in class, although we didn't talk about photochemistry? Yeah, benzophenone. Okay, and there's some other examples, so what's another example? Yeah, so they fall in here, right? So we can think about either just phenylazide, right, or fluorinated phenylazides. So another way to do this is to generate carbenes via diazarines here. Okay. And we'll pretty much focus on these types, which are major types. So where did this idea come from? Right? How new is this type of work, right? To stick a photoreactive group on a protein and then use it in a cross-linking application. And where did the idea come from, kind of in the first place? So what types of chemists often study photochemistry?
DNA, like
Well,
chemists, I guess.
more broadly, yeah. Yeah, so physical organic chemistry, right? There's a whole component of photochemistry there. Okay, let's take a vote. 2000? First photo cross-linker. 1990? 80? 70? 60? Just no clue? So around 1962 was the first paper using a photoreactive group on a protein here, Westheimer. And then Jeremy Knowles in 1969 was the first example of an aryl azide. Okay, so this work came out of physical organic chemistry and at a time where like physical organic chemists were transitioning into enzymology, right? Here, so we don't have time to go into a lot of the photochemistry of these different moieties, but is quite rich there. So how does this work? What types of reactions and groups get modified here in the cross-linking? So let's think about them. Okay, so let's consider an aryl azide. So what happens when aryl azides or azides are irradiated with UV light?
Evolve nitrogen gas.
Mm-hmm.
Get
Get
nitrine.
a nitrine. Yeah, so if we just think about nitrines for a minute, what types of chemistry did nitrines do? Are they reactive? So can they insert into CH bonds, NH bonds, add to double bonds? Can they do other things as well? Okay, so here we have our protein. What's going to happen? As Steve said, we're going to generate a nitrine. So how does that happen? We irradiate with light. Okay, to get our nitrine. So what happens with these aryl azides? is some interesting photochemistry when you're at a, say, room temperature. So rather than this nitrene reacting, say, with a CH bond or an NH bond, it actually undergoes a ring expansion. Okay, and so what we get, and this is very fast. So on the order of 10 to 100 picoseconds. So this happens before it has a chance to react with something else. Okay, to give us this intermediate. Okay, and so this species has very different chemistry than a nitrene. And what happens is it will react with nucleophiles. So imagine our amino group here to give the crosslink. So this pathway is the dominant pathway if just an aryl azide is used here. To think about from the standpoint of wanting to do cross-linking, so let's say you attach this aryl azide to a protein of interest, okay, and then you irradiate with light and look for it to cross-link with something. Is this an issue? It will form a crosslink. Would you rather have a nitrene react or this 7-membered reaction with this 7-membered ring?
I know you're saying that I would normally the nitrene, but it would depend on what you're actually looking at. It's like what you were investigating.
Okay, so why would you argue for the nitrene?
Well, like we were talking about, the nitrene does have a capacity to do like CH insertion or insertion or do NH. So if you wanted to do something like that kind of chemistry, then having this be the dominant pathway would be inefficient.
Yeah, right. There's a lot more CH bonds than there are lysines or N-termini, right? So that's one aspect. We've lost that chemistry. And then to another point, just as, how well do these reactions work? So nitrene reactions are very fast, relatively speaking. This is kind of sluggish there. And so, the question is, what can be done in order to improve upon this? Okay, and Steve mentioned these fluorinated phenylazides there. And so, photochemical work unrelated to any sort of, biological cross-linking chemistry showed that if you fluorinate aryl azides, you can get nitrene reactivity rather than this other pathway here. Okay, and so if we just take a look at that, what happens? Right, so for instance, imagine we have this tetrafluoro analog here. Right, we can imagine irradiating this and getting to our nitrene. I'm going to skip the steps. Okay, now what can happen? Imagine, right, we have some CH bond nearby. Okay, we get this crosslink. Okay, and this reaction is very, very fast. Here, very, very fast. So can ring expansion occur in this situation? Okay, so I'm pointing this out because the language in the packet was a little strong. If there's something for this nitrine to react with nearby, it will react, right? But this can undergo ring expansion. It's just much slower than the case above. So the studies I've read say about 170-fold slower there. So it's not that the pathway is completely blocked. And it also, too, depends on the experimental conditions. But anyhow, this is, quoted to be like near diffusion controlled here for that. So, this is pretty interesting when you think about it, right? Because azides, aryl azides, they can be fed to cells to do unnatural amino acid incorporation, right? They're used in click chemistry, for instance, types of conjugation chemistry. But here, the photochemistry can be taken advantage of, right, to give a crosslinker that can be controlled in a temporal manner there. So what about the benzophenone? What does benzophenone react with? After being irradiated with light. So imagine you have your protein, and maybe in this case, you did unnatural amino acid incorporation to site-specifically attach a benzophenone. What happens? What happens is that there's formation of a triplet diradical. Okay, and what will this do? Okay, this can react with some CH bond. Okay, to get the crosslink. So let's say you have this guy here, and you want to do a cross-linking experiment, right? So we can imagine some different possibilities. What do you think you'll get out? Are you only going to get out your desired crosslink? What might happen? To 150 on a Friday. Let's get some like jumping jacks, come on. Should I dismiss all of you? Because there's a major energy load today, I have to say. There, yeah. So do you think you'll get one product, 10 products, 100?
Lots of side reactions.
Yeah, right. There's still the possibility for many side reactions, right? And you always need to be aware of that. So if you cross-link something, right, the next question is this something that's actually relevant or not? Or is it an artifact there? So the analysis can be very complicated, right? And so that's just something to think about. Say you have cross-linked species from cell lysate. What are you going to do to analyze that? Right, so just think about some of the things that have come up in other contexts here, right? We talked about protease digest and mass spec, right, for looking at substrates of GROW-EL, GROW-ES. That's something that can be applied, and there's many sophisticated new tools to get a lot of information about out of the mass spec, which we won't talk about, but like having tags within the crosslinker, right? So then you need to ask how well is the coverage going to be, right? So even after this step, there's a lot more work, which we won't go into details in this recitation today. What about kind of inherent like efficiency of cross-linking in terms of these benzophenones versus the aryl azides? We want to think about relative cross-linking efficiency. Any sense of that? Mm-hmm.
I think the benzophenone compared to like the diazirin is a lot less efficient. I don't really know between these two though.
Mm-hmm.
Is, when we're talking about efficiency, is it purely based on the speed of this reactivity, or is it also taking into account the different cross reactions that could occur? Because it seems like there are more possibilities for cross reactions, even though it might be more reactive. It's not like, yeah.
The former, right? Just thinking about the reaction, right? There's the possibility of cross reactions for all of these. They're highly reactive. Like a nitrene is highly reactive. The benzophenone triplet diradical is highly reactive. A carbene, right? If you're going to get that from some diazirine, is very reactive. Here, I mean, is this something important to think about in terms of your experiment? What is the relative efficiency of the reaction, right? So I said that aryl azide's a little sluggish compared to the others. You know, something to consider, right? Do you know kind of what is the time scale of whatever it is you're trying to trap? So the wavelengths, what is it about these wavelengths that might be undesirable?
Well, combination of shift, well, for in vivo studies, one shifting towards UV means that you can have issues of, you know, undesirable like DNA cross-linking and stuff, but also it means that it's not going to have deep penetrance as you shift towards blue.
Yeah. So what wavelength would you like?
I would like it out around 650.
Yeah.
Now, these are all at use a UV visible interface, and you have hundreds of things that absorb light. They're incredibly inefficient. You get one hell of a mess. Most people never identify what they get out the other side. They just see two things stuck together, and that's the extent of it. They never describe the molecular details.
Right, so let's actually just close. Right, so one of the questions I asked in the discussion section is it worth the effort, right? If you're going to site specifically put in a crosslinker, and imagine you find this protein-protein interaction, right? If one chooses, you can do quite a bit more experiments in terms of where you place this crosslinker and mapping out you know, that interaction region there. And so that's, I think, also just a take-home is often you need to put, you know, your reactive group in more than one place to really get at the answer to the question you're asking. And so, you know, there's folks around doing that there, but, you know, is it 20 positions? Is it 10? Is it 50, right? Because if you don't know at the beginning, you may need to do a lot of just systematic trial and error for that. Yeah, so I think you should all read the packet. And there are some suggestions for reading, if you're curious to learn more, one of which is a manual from Thermo. So often the companies give a lot of like good general background information, and there's many different types of chemistry included in that as well. I'll also point out Ed is here for those of you who don't know Ed. So he'll be presenting next week on cryo-EM, and you should definitely read the fatty acid synthase paper beforehand. The structures are incredible, and fatty acid synthase serves as a basis for our discussions of polyketide and polyketide synthesis, which is where we'll begin module 4 in thinking about the biosynthesis of natural products there. Okay, have a good weekend.